Stem cells in regenerative medicine. Stem cell therapy represents a groundbreaking shift in the medical treatment through its potential in regenerative medicine. This therapy has been successfully applied in the treatment of various medical conditions, including autoimmune, inflammatory, and neurological disorders. Stem cell research is a rapidly evolving field with the potential to revolutionize the treatment of many diseases, including sickle cell anemia, Parkinson's disease, spinal cord injuries, type 1 diabetes, type 2 diabetes, heart diseases, stroke, burns, rheumatoid arthritis, Alzheimer's, cystic fibrosis, end-stage liver disease, ischemic diseases, various skin diseases, heart failure, liver cirrhosis, corneal diseases, and claims of many more. If the validity of these claims were unquestionable, why do regulatory bodies in the numerous countries, including FDA in United States, issue warnings and withhold approval for the therapies targeting numerous diseases? In this video, we'll discuss the fundamentals of regeneration, offering an introduction to stem cells. We'll explore the various types of the stem cells, their potential, and ethical considerations regarding their use. We'll also discuss safety concerns associated with stem cell therapies. Regeneration is a natural process by which living organisms repair or replace damaged or lost cells, tissues, or organs, restoring their structure and function. It ranges from biological mechanisms of healing of minor wounds and the regrowth of entire body parts in some species. Regeneration plays a vital role in maintaining health and life across various organisms. Understanding its mechanisms holds significant implications for regenerative medicine. Regeneration in plants. Plants possess remarkable regenerative abilities in various parts of their structures, enabling them to recover from damage or stress. They can regenerate from cuttings or fragments to develop into new plants under favorable conditions. Many plants can regenerate damaged tissues such as roots, stems, and leaves through specialized cells called meristems, which contain undifferentiated cells or stem cells, capable of continuous divisions and differentiation. Regeneration in lower animals. Regeneration in lower animals such as flatworms and salamanders show remarkable abilities to regenerate lost or damaged body parts. Particularly species like planarians exhibit extraordinary regenerative capabilities, being able to regenerate complete organisms from tiny body fragments. Their remarkable regenerative capacity is attributed to the presence of pluripotent stem cells, called neoblasts, which proliferate and differentiate to replace lost tissues. Similarly, salamanders possess impressive regenerative abilities, with some species capable of regrowing lost limbs, tails and spinal cords, and even parts of their hearts and brains. This remarkable step involves the activation of specialized cells at the site of injury, which undergoes dedifferentiation and proliferation to form blastema, a mass of undifferentiated cells, capable of giving rise to new tissues. In humans, regeneration occurs at a limited extent compared to flatworms and salamanders, but it still plays an important role in healing and tissue repair. The human body has some capacity to regenerate certain tissues and organs. For example, skin can regenerate to repair minor wounds. The liver can regenerate to restore its functions after injury or partial removal. Tissue regeneration primarily occurs through two main mechanisms: mitosis of existing cells and the activity of stem cells. Mitosis involves the division of existing cells to produce new cells, allowing for the replacement of damaged or lost tissues. This process is particularly prominent in the skin and epithelial tissues lining the surfaces of the organs. However, many other tissues in the human bodies have limited mitotic ability or lack the capacity for mitosis altogether. For example, nervous tissue, including neurons in the brain and spinal cord, typically do not undergo mitosis in adults. Similarly, mature muscle cells have limited mitotic capacity. In these tissues, regeneration primarily relies on the activity of stem cells. The regenerative capacity of stem cells in humans is not unlimited. Additionally, the regenerative potential of stem cells can decline with age or in certain disease conditions. Another difference from lower taxa is humans lack pluripotent stem cells in their body after embryonic stage. So stem cells have less potential compared to lower animals. Stem cells are undifferentiated cells with the remarkable ability to develop into specialized cell types and self-renew through cell division. They serve as the building blocks of the body, forming the foundation of all tissues, organs, and systems. Stem cells are characterized by their potency or their capacity to differentiate into various cell types, ranging from muscle and nerve cells to blood and bone cells. All lives start with the stem cells, the zygote with the full potential to develop into individual. Zygote forms embryo and extra-embryonic layers. Organogenesis is due to differentiation of stem cells in the embryo to form fetal body, so that stem cells with the variable potency remains in the various parts of humans throughout our lives. Types of stem cells based on their potential. Stem cells are often classified based on their potency, which refers to their ability to differentiate into different cell types. Here are main types based on the potency. Totipotent stem cells. The most potent type of stem cell. can give rise to all cell types in the body, including both embryonic and extraembryonic tissues, found in the very early stages of embryonic development, typically within the first few cell divisions after fertilization. Examples: cells of the zygote and early blastomeres. Pluripotent stem cells can differentiate into cells from all three germ layers: ectoderm, endoderm, and mesoderm, found in embryonic tissues. Offer great potential for regenerative medicine and tissue engineering due to their ability to become virtually any cell type in the body. Examples are embryonic stem cells, multipotent stem cells, can differentiate into a limited range of cell types within a specific lineage or cell types, found in various adult tissues and organs, play a crucial role in tissue homeostasis, repair, and regeneration within their respective tissues. Examples are hematopoietic or hemopoietic stem cells, found in bone marrow and capable of producing various blood cell types. Oligopotent stem cells can differentiate into few closely related cell types within a specific lineage or tissue type, similar to multipotent stem cells, but with a more restricted differentiation potential. Examples include myeloid progenitor cells give rise to different types of white blood cells. Unipotent stem cells, typically found in adult tissues and are responsible for replenishing and maintaining specific cell populations. Examples are spermatogonial stem cells and basal cells in the skin. Induced pluripotent stem cells. Induced pluripotent stem cells represent a groundbreaking innovation in the stem cell research. iPSCs are generated by reprogramming adult somatic cells, such as skin cells or blood cells, back to pluripotent stage, that is, to embryonic stem cells. This reprogramming is typically achieved by introducing specific combinations of transcription factors or genetic factors to the adult cells, which reset their epigenetic signature and gene expression profile to resemble that of embryonic stem cells. iPSCs share many characteristics with embryonic stem cells, including their ability to differentiate into cells from all three germ layers: ectoderm, endoderm, and mesoderm. One of the key advantages of iPSCs is that they can be derived from the patient's own cells, eliminating concerns about the immune rejection and ethical issues associated with the use of embryos. Despite challenges such as genomic instability and variability in the reprogramming efficiency, iPSCs continue to revolutionize the field of regenerative medicine, offering new hope for treating a wide range of diseases and injuries. Types according to source. Stem cells can be categorized based on their origin, which refers to the source from which they are derived. Here are main types based on the origin. Embryonic stem cells or ESCs, derived from the inner cell mass of blastocysts, which are early stage embryos. considered one of the most potent types of the stem cells due to their ability to form any cell type in the body. They are pluripotent, so differentiate into cell types of all three germ layers: ectoderm, endoderm, and mesoderm. Adult stem cells, also known as somatic or tissue-specific stem cells, found in various tissues and organs throughout the body, including bone marrow, brain, skin, and muscles. They can differentiate into a limited range of the cell types related to their tissues of origin, play essential role in tissue maintenance, repair, and regeneration. Two important types of the stem cells in the bone marrow include hematopoietic stem cells and mesenchymal stem cells. Hematopoietic stem cells, found primarily in bone marrow, but also in small quantities in peripheral blood and umbilical cord. They are multipotent and can differentiate into various types of the blood cells, including RBCs, WBCs, and platelets. Mesenchymal stem cells, found in various tissues including bone marrow, adipose tissues, umbilical cord tissues, and placenta. That is, they can differentiate into a variety of cell types such as bone cells, osteoblasts, cartilage cells, chondrocytes, and fat cells, adipocytes. Play important roles in the tissue repair, regeneration, and immune modulation. Fetal stem cells. They are derived from the tissues associated with the birth, such as umbilical cord blood, umbilical cord tissues, and placenta. Include umbilical cord blood stem cells, umbilical cord mesenchymal stem cells, and placental stem cells. Can differentiate into multiple cell types and possess immunomodulatory properties, making them valuable for regenerative medicine. Differentiation. Differentiation is a process by which cells become specialized to perform specific functions within an organism. This process involves changes in gene expression that leads to the production of specific proteins necessary for the cell's functions within the cell. That leads to the production of specific proteins necessary for the cell's function with the same set of DNA. Differentiation occurs during embryonic development as the cell undergoes various stages of specialization to form different tissues and organs, and is continuous throughout the life as cells replace lost or damaged tissues. At the molecular level, differentiation is governed by the regulation of gene expression. All cells in an organism contain the same DNA sequence, which contains the instructions for making proteins. However, not all the genes are active in every cell all the times. Instead, different genes are turned on or off in a coordinated manner to determine the cell's identity and function. Gene expression regulation. Certain genes are activated or expressed, while others are silenced or repressed, depending on the signals received by the cell from its environment. These signals can come from neighboring cells, developmental cues, or biochemical signals within the cell itself. In the cell cycle, differentiation typically occurs during the G1 or gap 1 phase. During G1, the cell is actively synthesizing RNA, proteins, and other molecules necessary for growth and function. It is during this phase that the cell receives signals from its environment that determines its fate and trigger the expression of specific genes associated with differentiation. Most human cells after completing the G1 phase enter a non-dividing state known as G0, where they remain differentiated throughout their lives. Cells in the G0 are metabolically active but are not actively dividing. This state allows differentiated cells to perform their specialized functions without undergoing further cell division. Cells in the G0 must be stimulated by appropriate signals to re-enter the cell cycle and progress through the subsequent phases, including S or synthesis phase, where DNA replication occurs, and G0 or gap 2 phase, where the cell prepares for mitosis. It's important to note that proliferation or cell division and differentiation are indeed opposing processes. This balance between proliferation and differentiation is tightly regulated and essential for normal development. Dysregulation of this balance can lead to the developmental abnormalities, cancer, or degenerative diseases. Complexity of proliferation and differentiation in stem cells. Stem cells have high proliferative potential, and when this proliferation is dysregulated, it can lead to the formation of tumors. This is particularly concerning in the context of embryonic stem cells and induced pluripotent stem cells, which have the capacity of unlimited self-renewal. Stem cells often express genes associated with the tumor angiogenesis, such as vascular endothelial growth factor or VEGF, which promotes the growth of blood vessels to supply nutrients and oxygen to tumors. The behavior of stem cells in terms of proliferation and differentiation is highly complex and dynamic. Stem cells can undergo asymmetric cell division, where one daughter cell retains stem cell properties while the other differentiates into specialized cell type. Alternatively, they can undergo symmetric cell division, where both daughter cells either retain stem cell properties or differentiate. The regulation of these processes is influenced by various intrinsic and extrinsic factors, including signaling pathways, transcriptional factors, and stem cell microenvironment. This inherent plasticity and unpredictability in the behavior of the stem cells pose significant challenges for their therapeutic use. Strategies to control and direct stem cell proliferation and differentiation are actively being investigated to mitigate the risk of tumorigenesis and improve the safety and efficacy of stem cell-based therapies. This includes optimizing culture conditions, identifying specific molecular markers, and developing methods for targeted differentiation into desired cell types. Risk associated with the stem cell transplantation. Immune rejection. The recipient's immune system may recognize the transplanted stem cells as foreign and mount an immune response, leading to rejection of the transplanted stem cells. This can occur particularly in allogenic transplantation, where the stem cells are obtained from the donor with a different genetic background than the recipient. Graft versus host disease. In allogenic stem cell transplantation, donor immune cells present in the graft may attack the recipient's tissues, causing graft versus host disease. This condition can range from mild to severe and may affect various organs and tissues, leading to significant morbidity and mortality. chances of infection. In case of allogenic transplantation, recipients are at increased risk of infections due to immune suppression. These infections can be bacterial, viral, fungal, or parasitic in nature, may lead to serious complications or death if not promptly treated. Graft failure. In some cases, the transplanted stem cells may fail to engraft and establish a functional hematopoietic or tissue compartment. This can occur due to inadequate conditioning, immune rejection, or other factors, resulting in the need for additional transplantations or alternative treatments. Organ toxicity. Conditioning regimes used to prepare recipients for the stem cell transplantation can cause toxicity to various organs, including liver, kidney, lungs, and heart. Tumor formation. There is a risk that transplanted stem cells or their derivatives may undergo malignant transformation, leading to the development of tumors or cancer. This risk underscores the importance of rigorous screening and quality control measures to ensure the safety of stem cell-based therapies. Some stem cell types may raise ethical concerns in different nations. This can lead to public debate and controversy surrounding the ethical implications of stem cell research and transplantation. Patients and their caregivers should carefully consider several factors before pursuing stem cell therapies or clinical trials. You should ensure that any stem cell therapy or clinical trial you are considering has been approved by appropriate regulatory authorities in the country. You should be aware of potential harms and risks associated with the therapy, including adverse effects, complications, and possibility of the treatment failure. If considering participation in clinical trial, you should carefully review the trial design, protocol, and inclusion or exclusion criteria. You should explore alternative treatment options and consider whether conventional therapies or other medical interventions may be more appropriate for your condition. How can you improve your own stem cells?